for today's SLAAM seminar. And it gives me great pleasure to introduce and invite Ido, who will be speaking to us today. So Ido Levin and his PhD from Hebrew University, working in the group of Eran Sharon, where he did lots of very, very cool work on both experiments and theory on all kinds of geometrically frustrated sheets, thin sheets, and the mechanics and the shape and how they change shape and how they have anomalous mechanics, all due to geometric mechanisms. He is currently a Fulbright Fellow and also a Washington Research Foundation Fellow, working in the University of Washington with Professor Sarah Keller's group, working on various other problems, related problems, but on a very different scale, of shape-morphing structures and understanding the interplay of geometry and shape and mechanics, but now in the context of biological membranes, lipid membranes, and smaller scale vesicles. And so we're very excited to hear more about what you've been up to. And so take it away. Small minor comment for the audience. Ido has kindly agreed that people should feel free to interrupt and ask questions throughout the talk. But if you don't feel comfortable with that, feel free to add your questions in the chat and we will monitor it and interrupt accordingly. There'll also be time at the end of the talk for Q&As at the very end. So we'll stay on. Thank you and take it away, Ido.
All right, so thanks, organizer, for the introduction and for inviting me to tell you today about 3 projects I was involved in. And these will be 3 distinct projects, but they have a common underlying theme, which is trying to understand better shape-morphing sheets, also improving our way both to model them theoretically and to be able to fabricate more complex shape-morphing sheets. And the motivation from such works come from nature. When we see many soft structures that have no rigid underlying structure, but nevertheless manage to change a shape in a quite striking manner. And here, for example, we see a beating human heart, and this is one of the most remarkable soft machines. It has, again, no rigid parts, but nevertheless managed to pump blood, and we all rely on its robustness when we sit and here today in this meeting. Another example, here it's a very primitive organism called a T-plax. And a T-plax is just 2 layers of epithelial cells, and we see contraction fronts propagate in these tissues and change the shape of this organism here laterally, so only in plane. And another example is this swimming nudibranch that does that so gracefully in the ocean. And these are just 3 examples. So there are many, many other examples of soft structures that manage to shape themselves. And if we try to understand the basic mechanism of these systems, and these systems are very different, and if you ask biologists, they will say there's so many different details between them, but as physicists, we'll try to distill the essence of what's common to these systems, so we can write or think of a very simple model that will explain what we see here, in which we have a propagating signal. So in the context of the heart, this will be the action potential fronts. And this propagating signal causes local deformation. So in the context of the heart, the contraction of the cells, which will give us the evolving shape. So we have some local information dictating a global change of shape, and we have some interplay here between this inelastic deformation, which can have many different sources. It can be contraction of cells, it can be growth, swelling of, for example, a hydrogel, local reorientation of elements, and many other sources, and elasticity, which controls the shape. And a quick outline of what I'm going to talk today. So briefly, I'll start with telling you a bit about this theoretical framework of how we understand these shape-morphing structures. Then I will tell you about a new pathway we discovered to shape-morphing that gives rise to different mechanical properties than what was known before. Then I will dive into the main project I will tell you today about, which are the autonomous oscillating membranes we work hard on and we are very proud of. And finally, I will, if time permitting, I will briefly tell you about an ongoing project in which we'll try to utilize 3D printing techniques in order to really program the shape of structures in a manner, in a fully controlled manner. And all these projects took place at the lab of Eran Sharon at the Hebrew University. The first project is a collaboration with Emmanuel Sifer, then at Huji, now the University of Brussels, Maor from the mathematics department. The second project is a close collaboration with Robert Digan from the University of Michigan, and now a new grad student, Omri Cohen. And the last project is a collaboration with the Shlomo Magdassi Lab, and especially with two students, Rabba Lieberman and Elach Sachiani. So as I told you, the key essence here is to separate the inelastic process from the elastic relaxation. So let's imagine I have this magic material that just by coloring it, I can induce inelastic deformation. So orange pixels will shrink and green pixels will swell. Just by coloring these two specimens in different colors, I already induced what we call the reference geometry. Now these structures have preferred rest length, elastic rest length along them. And different coloring will induce different reference geometries. Now I release these structures and let them adopt a shape. They will adopt different shapes. And these shapes will have the actual geometry of these adopted shapes. And we're working with hyperelastic materials in which the elasticity is derived from an energy. And we have some general strain, which measures deviation of the actual metric from the reference one. And in a moment, I will elaborate more. So now I will tell you the gory details. So if you're,
Can I ask a
you don't want,
here?
Yeah, sure.
Are you interested in the time course of the growth of the reference metric?
So this is a great question. So in the first part, I will not. I will assume there is some coloring field or a reference metric. But yes, in the oscillating membranes, the dynamics of these reference fields will be very important. I do assume there is some separation of time scale in which the reference state is well defined. So I can define the reference state, it can evolve, but elasticity reacts faster. So every time there is a shape, and then there's a new reference state, and there's a new shape, and so on. But there's a lot of interesting physics about the coupling between the propagation of this reference state and the elasticity. Yes. Okay.
Thank you.
Sure. So this is going to be a bit technical slide, so you can go and make your coffee if you're not into a very technical slides. But in practice, what we need to do, so we have our material, and we're working in a shell theory. So we approximate the reference geometry just to second order. So we have the distances at the top layer and the distances at the bottom layer of this plate or shell. And these distances give us what we call a reference metric, just a tensor that measures distances, reference distances in this material, again, caused by the coloring of it. And the actual reference metric of the material, it will be just the average of the top layer and the bottom layer. Differences between the layer will result in reference curvature, tendency of this sheet to bend. And this also given in this reference curvature tensor B bar. As you can see here, the difference between the two metrics, and H is the thickness of the sheet. And the actual configuration has an actual metric and an actual curvature tensors of the configuration itself. And then our elastic energy will be composed of stretching term, scales linearly with respect to the thickness, and measures deviation of A from A bar, and a bending term that scales cubically with respect to the thickness and measure deviation of B from B bar. And this seems quite straightforward. And you might ask yourself, okay, so where all this complexity of shapes that you see comes from? Just set A equal to A bar and B equal to B bar, and that's it. But actually, stretching and bending are coupled. And this is something that is well known to someone who likes to eat pizza. So if you try to eat your pizza and your pizza has some heavy topping, so your pizza might bend and ruin your pants. But if you buckle your pizza in the perpendicular direction, it becomes way more rigid in this direction. And this idea was formulated by Gauss almost 200 years ago. And Gauss told us that the Gaussian curvature, which is the product of the two principal curvatures along a surface, is an intrinsic invariant. Or in other words, in order to change the Gaussian curvature of a surface, you must stretch it. And this tells us there are some relation between the metric and the curvature tensor. So K can be expressed using the metric A alone, but it is also expressed using curvature, so the ratio between the determinant of B and A. So this gives us some selection rules between A and B. So the actual geometry is constrained. The reference geometry just results from this coloring scheme I showed you before, so it's completely arbitrary. And in cases where the Gaussian curvature K bar, which we get from calculating it directly from A bar, the reference metric, is not equal to the ratio of the determinant of B bar and A bar, we'll have geometrical frustration, which means no surface will have both A equal A bar and B equal B bar at the same time. In cases where A is not A bar, we'll have stretching, and B is not equal to B bar, will involve bending. And then the shape will be determined by a competition between stretching and bending. And this can give rise to a lot of complexity. However, in the thin limit, you see that the exponents of the thickness h are different between these two energy terms. So at the thin limit, stretching becomes very expensive. And in this limit, we expect A to be equal to A bar and K to be equal to K bar. And this is a way to program Gaussian curvature into flat sheets. And this idea was realized in many experimental systems using mostly responsive materials. And one of the first ones and the most versatile ones are hydrogels. So hydrogels can react to many external stimuli. It can be temperature, change of salinity, change of pH. You can shine lights on them. You can shine light on them. You can change the solvent and even dry them, and they can react to these external stimuli. And here we'll see one example. This is polynipam, a hydrogel which reacts to temperature. When we heat this gel, it shrinks. And as you saw before, it will shrink non-uniformly. And like that, we can program a reference geometry. So the inner part of the gel here is more diluted. It will shrink and collapse more. So the outer part will have excess length, and we'll have these hyperbolic configurations. And when we heat it up again, and when we cool it down again, it will just relax back to its initial state. And just by programming different lateral geometries, we can program different shapes to our gels. We can use more advanced techniques such as shining UV light to selectively cross-link our material. We can use 3D printer printing gels with some metal, with some glass or cellulose fibers in them to induce, again, reference geometries. Other researchers prefer to work with elastomers that are more rigid. So we have these pneumatic elastomers that can be actuated using light and heat. And we have these pneumatic baromorphs that can be actuated using pressure, the electric elastomers that can be actuated using the electric field, and there are many, many other ways to do that. And among all the things we can program and make, one very important class of structures are ribbons. So ribbons are structures, thin sheets, which we have another separation of length scales. So the length is much larger than the width W, which in case, again, is much larger than the thickness H. And there are many examples of frustrated ribbons, and we can make them from many different materials. So rubber, hydrogels, you can see self-assembled amphiphiles, metals, 4D printed wood, ceramics, and we recently even started to make biomimetic pasta. So pasta, which is frustrated. And when you try to understand the mechanics of these structures, all of them are frustrated due to the Gauss theorem I graded. And this gives us some very distinct properties for these ribbons. So first, they exhibit a very sharp shape transition. So you can see this one has like this twisted structure with Gaussian curvature. And when it becomes wider, it goes to this flat cylindrical helix. So all these structures exhibit sharp shape transition. In the thin, which in ribbons is also the wide limit, A equal to A bar, as we said before. In the thick or narrow limit, B equal to B bar. And when we try to understand how the critical width scales with respect to the thickness, it always scales with the square root of the thickness H. So we have like a universality class that all the frustrated ribbons we know of fall into. And then we thought of, yeah, sure.
Can I ask a quick question about the critical width?
Sure.
Is the square root H scaling the same as the boundary layer thickness, the thickness of the boundary layer that you get in curved shells?
So it is, but it's not exactly. This is like an Euler buckling. Okay. So you write the stretching and the bending energy, and just by equating them, we'll see it because I will show the same mechanism when we do like in a different, like in the other class of frustrated structures. And when you write it, you equate the bending and the stretching. So you have only stretching in the narrow limit, only bending in the wide limit. And in the transition, you just equate the two scaling laws. And this is what you get for this kind. And this is exactly what you get for Euler-Buckley.
Okay. Okay.
I hope it was. Okay. And then we thought of a structure which seems rather trivial. This is a ribbon. It is bent, so the midline is just curved. It is metrically flat, so the metric here is just the flat metric in polar coordinates. And it has just a radial curvature everywhere. So everywhere there is curvature at the radial direction. And you can immediately convince yourself that the Gaussian curvature associated with A bar is 0, because this is the flat polar metric. But also the determinant of B bar is 0. So there's apparently no frustration. And if there's no frustration, it's even easier. So what's going to be the shape? So you might try to connect all these radial curved lines into a torus. But the torus is non-flat. So neither A equal A bar or B equal to B bar. Then you might say, okay, it might straighten out. And then you have all the curvature in one direction. So this has no Gaussian curvature, but this involves stretching. A here is not equal to A bar. And we were pretty confused by that for a while. And then we said, okay, we're experimentalists, so we can just make it and test. So there are two ways to make such a ribbon. The first one is using pre-stretched rubber. So we take this pink rubber sheet, we stretch it radially. We added these lines to avoid the Poisson effect. So really, we had just radial stretching. And then we glue on top this green unstretched rubber, and we cut it. The other way is to use 4D printed thermoplastics. So these thermoplastics, when they're 4D printed, the 4D printing involves a very violent extrusion process. So the molecules are extruded along the printing direction. And when we throw it into a hot bath, then thermal fluctuation just shrinks the molecules back, and we get shrinkage along the printing direction. So we print two layers. The first one is this Hilbert pattern, which is just stuck in place. And the top layer is just radial lines. And again, when we throw it into a hot bath, we get the same reference geometry. And when we do that, we get this very faceted and nice structure. So we had our answer. But then we had an idea, okay, let's try to cut it now, reduce the width, and see what we find. And when we started doing that, we immediately saw that this ribbon changes its shape. And this is exactly what we did with the 4D printed thermoplastics. Here we just printed narrower samples, we cannot cut it. And also in numerical simulations. And this is quite surprising because these ribbons are not frustrated according to Gauss, but nevertheless their shape depends on the width. So this indicates they have some excess energy or residual stresses in their configuration. And this is the hallmark of geometrical frustration. So what kind of geometrical frustration we have here? And then we remember that there are other compatibility conditions between metric and curvature. And this calls the Mainardi-Codazzi-Peterson equations formulated by Peterson, which in very nice words told us that the normal to a surface behave nicely. And this means that the second covariant derivatives of the normal commute. And because gradients in the normal are expressed by the curvature tensor, and because these are covariant derivatives, you can take this condition and write it as a condition of a symmetry condition of the covariant derivatives of the curvature tensor. And when we plug in the reference geometry of our ribbon, and I'll skip the tedious calculation, we immediately see that we don't get this symmetry condition. So this ribbon violates the Mainardi-Codazzi equations. And here we identify a new type of frustrated structures. What we call, they have normal mismatch instead of Gaussian curvature mismatch. So the next step is trying to understand, okay, but do they all belong to the same universality class? Maybe they have the same scaling and they behave very similar to what we saw before. So we start with the thin limit. The thin limit is always easier. As I told you, it's always the case that you get A equal A bar at the thin limit. And we already saw the solution, but we are looking for developable surfaces. And what we saw from our experiments, that each part of this ribbon is just curved at uniaxially, at the same direction. And again, involving no Gaussian curvature. And then we try to understand how many facets do we have. So each of these bends, just we start and along, or this is, for example, our structure, and then it can bend uniaxially, but the maximum area that can bend uniaxially is just limited by this blue line you see here. And then we can add another and another, and then we have the leftover. And this tells us that the number of facets is just given geometrically by the ratio between the width and the radius of our ribbon. And we had this prediction of no fitting parameter prediction of the number of facets. And when we plug in our experiments and simulations, they work surprisingly well. And then something very nice that we also saw in this limit is between these two bands, we get stress focusing. So these sharp corners involve stress focusing, which is something that is rarely found in unconstrained sheets. And then we move to the bit harder limit, which is the thick or narrow limit. And our intuition from the Gauss frustrated structure is that B should be equal to B bar, which means straightening a ribbon into a tube and just bending uniaxially like that. But it turns out that this involves huge stretching energy. And actually the favorable configuration is a toroidal one, which again involves both stretching and bending because it is non-flat. And something interesting about these toroidal shapes, they have over curvature. So if we start with the 2 pi, these toruses just goes like that, and we have more and more total angle. And again, this is prediction has no fitting parameters, and when we plug in our experimental and numerical data, it fits very well. So now we understand both limits. We can also write the energetics of these two limits. So we have the scaling of energy at the thin limit. We have the scaling energy at the thick limit. And when we equate these two, we can find our prediction for the shape transition. And as you see, the transition that we predict has an anomalous scaling. So it's instead of the square root of h, it's like the third root of h. And our data agrees with this prediction. So in essence, we find a new class of frustrated structures. Another interesting thing to note is that the transition here is much wider. So usually in Euler buckling, you get a very sharp transition. It's either buckled or not. But here we have this orange wide intermediate domain region that does not appear in Gauss frustrated structures. So I will briefly summarize. So we identified a new type of frustration, which involved an anomalous narrow limit because it has both stretching and bending. But then we asked ourselves, okay, so does all the structures or ribbons frustrated according to the Mainardi-Kodasi equations have the same scaling? So do they all fall into the same universality class? And then we had a project that I will just summarize the details. I won't go over the calculations, but it turns out that once you allow also Mainardi-Kodasi frustration to appear, you can tune your scaling law to get any fractional or not like square root of the thickness for the transition. So we can get the square root of h, we can get the third root, the fourth root, the fifth root, and so on. Something also very interesting that there are frustrated structures that are frustrated but does not exhibit shape transition. So they remain frustrated both at the thick and the narrow limit, but for example, these conical shapes, they remain conical in both limits. And our method of trying to utilize the fact that in ribbons, the width is another small parameter. So we can expand both the energy and the geometry as powers of W. So in this model that we can go order by order, we can account for any arbitrary geometrical frustration and find if there is a shape transition, and if there is, what's going to be the power law. So this we're pretty excited about. And now I will very sharply move to the second. Yeah, sure.
Before you go to the next.
Yeah. So there's a chat. Perfect.
There's a question in the chat. Andre, do you want to ask it, or I can read it out? Okay, I can read it out. I mean, So Andre Zakharov asks why there is no azimuthal stretch when we pull radially. Presumably the circumference has to change too. And I'm not sure.
What?
I'm not sure.
I wasn't sure. So let me.
I don't know
like why do we get this reference? So is the question is why do we get this reference geometry? Like why do we have only radial curvature? And this is because the way we design this experiment. So we have these slits we introduced in the rubber. So when you stretch it radially, you avoid the Poisson effect. So it's true that usually when you stretch a material in one direction, it can shrink in some exotic material, even expand in the perpendicular direction. So to avoid that, you can think of it as many rubber filaments that just stretched in one direction. They are like 1D ribbons. I hope this answered the question. I wasn't sure that this was the.
Thank you.
Great. Yeah. Okay. All right. So then you can say, okay, this is good and well, but when you showed us these cool movies in the beginning, you had these nudibranchs that swam in the ocean, but now you talk about rubber. So there's quite a difference between these two. And indeed, in all the realizations that we manage and many other also can do, the actuation is global and static. So we can heat our gels, they can change the shape. Some of them are reversible. We can cool the gel back down and they go back being flat. But this is very different than what we saw before. And in this project, the goal was to try to actuate our sheets intrinsically and also autonomously, just let them go by themselves. And as a cartoon, if you show many researchers a picture of this nudibranch, there are many different techniques to take a flat disc and mimic the shape of the nudibranch. But this is very different than having something like that. Sorry. So how can we even start? So what the key components we need in order to get these swimming nudibranch? So first we need a shaping mechanism. And a shaping mechanism, as I told you before, involves this local deformation and the translation of the local deformation into the global shape. And in the context of the heart, again, this is the contraction field. Then we need, some orchestrating controlling signal that will change both in space and time. It will be like the action potential of the heart. And finally, and this is the hardest step, we need somehow to couple these two. We need this orchestrating signal actually to dictate the mechanical state of our tissue. This will be like the metabolism. We'll convert this electrical or chemical or whatever energy into mechanical work. And in this project, we chose to work with a very famous pattern-forming reaction as our actuation signal. This is called the Boleshov-Jabotinsky or busy reaction. It was one of the first autocatalytic reaction that was discovered at the 50s. And in this reaction, we have a metal catalyst, in this case, ruthenium, which is reduced and oxidized periodically. And you can see many nice movies online. When you have a beaker with a solution, the solution also changes its color. So it starts as orange, turns green, and then orange, and it can have many different oscillations. But if you run this reaction in a medium which is governed by diffusion, such as in 2D or inside a gel, gradients in chemical compositions will translate into evolving shapes. So this is a very famous pattern-forming reaction. And we get target pattern with just concentric spirals, concentric circles, that goes out, and spirals. And there is a variation of these polynipalm gels I showed you the movie of before that reacts to the busy reaction. And by reacts, I mean that in the green oxidized phase, it swells, and in the orange reduced phase, it shrinks. So this one is a very good candidate to be for our self-oscillating sheet. So it took some time, but we managed to synthesize large sheets out of this busy gel, what's called. And this is this orange disc. And this orange disc is inert. You can keep it in the fridge for weeks. But when you throw it to a solution of what we call the busy solution, which will be the food, will fuel this reaction, this is what you see. So you immediately see these green fronts. I hope you can see it well. These green fronts that propagate along these discs, and they are accompanied by a buckling out of plane. And when we saw that, we were very excited because this is the first example of a completely autonomous synthetic shape-morphing membrane. So the next thing is to try to see if our model can capture what we see here and trying to understand better this model. I'll just note that this is 200 times faster than real life. This reaction is quite slow. So if we try to understand this reaction better, we can just look at snapshots. And luckily for us, the state of the reaction is also changes the color of the gel. So we can just probe the state of the reaction by taking the green RGB channel of these images. And we see here snapshots of a different gel. This one has a radius of 10 millimeters, thickness of 500 microns. And the time difference between consecutive images here are 80 seconds. And we see a single target pattern that propagates. And now we can extract the information about the busy phase, the phase of the reaction along these colored lines. And we can see that the reaction is separated, or the dynamics is separated in two stages. In the first stage, we have a front that propagates more or less at the fixed velocity of 100 millimeters per hour. And in the second state, we have relaxation of the global, or the pattern into the initial state. And because the reaction takes place only inside the gel, so we don't waste fuel in the bath, so the fuel is wasted just inside the gel, this can last for many hundreds of oscillations. So because it is slow, I could start the reaction on Friday and go back after the weekend and still have a gel that keeps beating. And because we don't find any changes between the top and bottom layer of the gel, we expect no curvature, no reference curvature to be apparent here. So these gels will be controlled by reference metric. And so then we would like to measure the shape. And measuring the shape is a bit harder than measuring the reaction here. Because the gel changes its shape quite a lot, we need to work in Lagrangian or material coordinates. So I put these black markers on the discs, and I take pictures using two tilted cameras. And then I identify these markers with one camera, I identify them with the other cameras. And from the difference between these markers, we get the Z coordinate. So just a stereoscopic apparatus, like our eyes manage to reconstruct 3D. Then we can also track these particles using particle tracking to get the dynamics. And again, once we have these Lagrangian coordinates, we can project the color field on them and get the busy phase in these Lagrangian coordinates. And this is how it looks. So on the left side, you see the raw image of a beating busy gel, and on the right side, you see the beating or the reconstructed shape from two different projections. And you can immediately see that the shape changes quite a lot, and also that the sign of the Gaussian curvature changes. And if you remember what I talked about 20 minutes ago, the Gaussian curvature is a very important mechanical property. So the fact that we managed to change the sign of the Gaussian curvature indicates that these local deformations are quite large. So now we would like to try to do it more quantitatively. So this is the reconstructed Lagrangian coordinates on one gel sample. And now we can zoom in at a specific vertex in these coordinates, and we can track at the neighboring triangle, both at the busy phase, just the colored, which is this red oscillating signal, and also the relative area changes, which is this blue oscillating signal. And these two signals are highly correlated. There's some time delay of 80 seconds. And if you try to understand this time delay, this is just the time it takes the gel to equilibrate. So if we take a piece of gel and throw it into an acid, for example, it shrinks very rapidly. But what do I mean by very rapidly? With a time scale of 80, 85 seconds. So this is just the time it takes to expel water from the gel matrix. Then we can ask ourselves, what kind of deformation do we find? So we can look at these two histograms. So the blue histogram just tells us for each triangle how much its area changes along this deformation. And the red signal tells us by how much the aspect ratio or angles changes. And while we do find a strong signal of change in area, we don't find a change in aspect ratio. And this is what we expected. These gels are isotropic. There's no preferred direction. There's no reason to believe the triangle will just get elongated in one direction, just swollen and shrunken uniformly. So once we know that, we can try to get the constitutive law. So we can correct for this 80-second delay, and we get the growth as a function of the busy phase here. And we fit this linear trend, and this linear trend is fitted for the entire data from this gel, not only the specific patch you see here. And because we know that the growth is isotropic, so circles just grow and shrink in area. This is how the reference metric evolves in time. So the reference metric is just the flat metric times this factor omega, which depends on the phase of the reaction. And when we have this form of metric, calculating the Gaussian curvature is quite straightforward. So the Gaussian curvature, in this case, the reference Gaussian curvature, involves second derivatives of the logarithm of this omega field. So in this scheme of propagating signal that control local reference geometry and then shape, I showed you that our system, we understand well the first arrow. So what's about the shape? So Something very interesting about this geometrical formulation of elasticity is that in many cases, we can avoid hard calculation. And as physicists, we're pretty lazy. We don't like to work hard. And in cases when we can exploit geometry to avoid calculation, we try to do that. And this is a very nice example of it. So this is a snapshot of our gel. And you can immediately see that the transition between these two domains is very sharp. So we can model these as made out of two areas, a green area and an orange area, uniform ones, with a circular boundary between them. And the green area is just swollen with respect to the orange one. And now you try to think, what will be the shape? So because these two domains are uniform, we expect them to be flat. There will be no Gaussian curvature because Gaussian curvature relates to the metric with a second derivative. So if we look along this line, we'll expect zero Gaussian curvature, then a fast oscillation, which involves second derivatives of a step function, and going back again. So how can you make such a surface? So one guess will be trying to glue two cylinders together, which is almost the solution here, but the boundary between two cylinders is a straight line, and here we need a curved boundary. So a small correction to that, just gluing two cones together, will give you the amount of degree you can to control the radius of this boundary line. So actually, we predict that in this geometrical or thin limit, the shape of our gel will be just a disk cut from this structure made out of gluing two cones together. Then we can look up, at our data, and we can also run numerical simulations. And here we can get much better resolution. It's easier to increase the resolution in our numerical simulations. But you see that the simulation. Sure.
Ido, sorry, can I ask a quick question? So when you say glue two cones together, what's your gluing procedure?
So that's a good question, and this will involve the specifics of this, how exactly the boundary looks. But you can assume, for example, a hyperbolic tangent or something like that, and then you just smoothly transition from one to the next. It's true that the exact details of how this function oscillates at the boundary will depend on this gluing process, but far away you won't feel it. Far away you're flat. And the angles of the cone is fixed because it tells you the radius of this boundary. So the details at exactly here are important, or they depend on the exact gluing process, but far away we don't really care.
So you don't require that the gluing, you don't worry about the mechanical consequences of the gluing as of now. You're just using this as a
just getting the shape. So it's not really gluing. It's just like you try to construct a shape which is flat on one end, flat on the other hand. It has one is swollen with respect to the other, so it must involve a jump in Gaussian curvature, because if you look at a conformal factor and you take a second derivative, you have a jump in Gaussian curvature. And again, it's true that if you really try to fit this red line, so you'll have to find the right transition or right, gluing process, let's call it. But if you care only about this flat and the fact that they are, the conformal factors are different and that it's like that along a curved line, so you don't really care exactly what happens in this transition, in this region.
Okay, thank you. And I
sure.
have a question on asking about if there's any chemical gradients across the thickness.
So we couldn't detect any chemical gradient. So our way to probe the chemistry is just the color field. So we have no chemical probe inside, but when we took pictures from the top and bottom, they look the same. I had discussion with people working on busy gels, thicker ones, and they told me that in 500, this is more or less, in 500 microns thickness, this is more or less the limit when you expect no, or at least not significant differences between the top and bottom. But it's true that if you'll take thicker gels, especially, of course, thicker is with respect to the length scale of the reaction, you can change it a bit by changing the chemistry. But if you fix the chemistry and you just make these gels thicker, you might get fronts that will behave differently at the top and bottom. And they also might propagate not at the lateral direction. They might propagate also in the Z, like the thin dimension to some extent. So it's true that when you make these gels thicker, it's not necessarily true, but we could not detect differences in this case between the top and bottom. And we also work out that the gel's composition will be the same. You can also make gels which are asymmetric, and then you break this symmetry.
Presumably, if you put the sheet near a surface, or if you supply fuel only from one side, then you can play with differential chemical gradients across the thickness. Is that correct?
Yeah, definitely. And also, if you put the gel next, like near a surface, you have many or much more richer physics, because then surface tension will play a role. The gel cannot buckle because it needs to get water out of the fluid. But it's true that by placing it at the surface, and there are some works trying to understand the relation between surface tension and these busy gels. But here the gels are submerged in fluid, so they are all around them, and they are covered in we assume more or less the same composition of chemicals. And again, we couldn't detect. We worked pretty hard to try to break the top-down symmetry. I hope it was easier than it turned out to be, but we couldn't detect.
Okay, and Andre has one more question on why should we be thinking of gluing cones or cylinders if the two sections of the sheet are flat?
So cylinders and cones are locally flat. So cylinder is completely flat. Cone, it's true that a cone is a result of a Gaussian curvature source at the tip. But if you cut the cone, you can take a piece of paper, and I can take this piece of paper, which is flat and unstretchable, and make a cone out of it. And so if we try to look at developable surfaces, which are so cones and cylinders are good candidates for that. And also, it turns out that you need this degree of freedom to be able to calculate, to account for this varying radius. So when time evolves, this front will propagate, and the radius of the boundary between the two domains will change. So you need somehow to be able to account for that, and cones gives you the amount of freedom you need, and you don't need more than that. So it turns out that this is a good guess. And as you'll see in a second, it also seems to work fairly well.
Thank you.
All right. So to show you that it works fairly well, we also ran numerical simulations. And you see that they look very similar to what we had in experiment. But while they resemble what we see here, this one has a very sharp transition, while this looks much more smooth. And we try to understand what happens here. So from the numerical simulation, we can look at the Gaussian curvature at a very high resolution. And when we look at K bar, we have a very sharp oscillations, and we saw before, and we have a cutoff here just so we can see K. So this is why it looks like a uniform blue and red, but it's just sharp oscillations with a very high amplitude. But K is more spread around. So the Gaussian curvature cannot account for this high oscillation of K bar, but it's spread around. And if we try to understand that, so instead of having these reference Gaussian curvature oscillating up and down rapidly, we have more smooth actual Gaussian curvature. And in this case, when we try to smooth this transition between the two cones, we get configuration which looks very similar to what we get in the finite element simulation and experiment. And this is something that was nice in this case and in many others, that from geometrical argument, you can not only get the geometrical or thin limit, you can also account for small finite thickness effects. So it's true that if you go very far from the thin limit, geometrical arguments won't work. But here we understand the first corrections to the thin or geometrical limit. But we can even do better. We can really try to measure the Gaussian curvature from our experiments. We already know that K is smaller than K bar, so actual geometry is not the reference one. And again, this involves some bending, some finite thickness effects. And if we look at the literature, we find theoretical works that dealing with similar reference geometries. And when we plug in our experimental parameters, we find that K should be more or less 20 or 25% of K bar. So we can measure the reference Gaussian curvature just from the color field, again, taking this second derivative. We get this oscillating signal for the same patch you saw before. We can, from the shape, reconstructed shape, measure the actual Gaussian curvature, and we get this oscillating red signal. And when we put them one with respect to the other, we see that we have two regimes. In the first one, K indeed more or less 20% of K bar, which is what we predicted. But in the other one, we have a sharp cutoff, which we were pretty worried about. So we try to understand what is this sharp cutoff. And when we try to plug in the numbers, so this sharp cutoff in K, in the actual Gaussian curvature, corresponds to radius of curvatures at the boundary or the transition between the two phases, which is more or less twice the thickness of the material. And when you try to understand what's the maximum value of the reference Gaussian curvature, it corresponds to radius of curvature, which is at the thickness, even smaller than the thickness of the material. And this is something, of course, that we don't expect our material to have such sharp corners at the scale of the thickness. So this is why we have crossover between these two regimes. So mechanically, we understand what happens here, but we can do even more with these materials. So it turns out we can control these materials using light. So blue light suppresses the reaction, and our collaborator Robert Digan built this Galvo mirror apparatus where he can trace blue laser, into arbitrary patterns, evolving pattern. So he can erase the initial spontaneous pattern in a busy gel and program a localized spiral. So here we'll see a movie. There will be an initial spiral around this X, then there will be a blue mass of shining the laser, and then we'll have a long-lasting spiral around this point. So let's see the movie. So you see this is the original spiral. Now we erase the pattern completely, and we program a spiral exactly where we want it to be. So we don't have to, but in cases we like, we can jumpstart the system in a specific pattern we would like. And something else we are very interested in, and I already hinted before, is motility. So what about swimming? You showed us these cool movies. You talked about the nudie branch. So far, the gels just changed their shape, but they were stuck at place. And this is something we work hard on, and it's a collaboration now with a student that continued the work, Omri Cohen at the Eran Sharon's lab. And what we do here is we look at ribbons, so elongated configuration, not disks, and we try to see if we manage to get them to move. And sometimes they do, and sometimes they move very nicely, such as this gel. And here we just track the center of mass of the gel. Sometimes they do not a very good job, and we get a population and more of such swimmers. And when we try to understand what is the swim velocity, is 10 millimeters per hour, which is not great. Non-Olympic records will be broken here. But if you take into account the fact that the front velocity is 100 millimeters per hour, it's not too bad. And now we are trying to understand these swimmers. So the first thing is to see in which regime they live in. And when we plug in the Reynolds number, which tells us that is it a laminar swimming or a turbulent swimming, we find that the Reynolds number are more or less 10 to the minus 2. And just a reminder, if you're not familiar with Reynolds number, so at the large and fast scale, we have, for example, a fish that swims, or even a whale. This is 10 to the 9th or 10 to the 5th Reynolds number for the fish. Bacteria, which are very low Reynolds number swimmers, are 10 to the minus 4. And our gel is pretty close to these bacteria. And in this regime, we have what's called the scallop theorem. The scallop theorem tells us that in order to get swimming, we must have irreversible deformation. So a scallop that does just that will propagate forward and then backward and then forward and then backward. And in order to get real net propulsion, you need to have some irreversible deformation. And people at the soft swimmer community work very hard to get and then get these irreversible deformations. But here we just get it for free for the form of the propagating reaction in our sheet. So this is something nice. So Omri now is teaching these gels how to swim, looking at gel fluid interactions. And something interesting that he also noted is when you put several swimmers together, you get this very interesting synchronized swimming that takes place, which now we also look into gel-gel interactions. So I have, I think, few minutes left. So I will very briefly tell you about a new project. We're still, sure.
Ido, before you go ahead, there's one question from the chat by Jan. How is the BZ catalyst bonded to the hydrogel, chemically or sterically?
So it's chemically, it's like part of the backbone of the gel. So that's true. I haven't mentioned that. This is like polynipam, but it's a double, like we have another monomer, which is part of the backbone of this gel, which include this ruthenium center that reacts to the BZ reaction.
Thanks. And Tim Bilash has a question on what happens if you put solid islands dotting the disk?
Solid island, like if I take a BZ gel, or I don't need a movie, but a BZ gel and put something which is passive, or what do you mean by solid island?
If the gel is not uniform. So think of little Pacific islands.
Yeah, so this is another great question. And we worked hard on trying to find ways to do that. This is one of our ideas of trying to control the reaction, having different domains, for example, with different propagation speed, maybe some domains which are completely passive, have no ruthenium. We started working in this direction, but still very preliminary. So we are very interested in this question. Hopefully we'll have answers soon.
Okay. Okay, I think you should go ahead because you have about 10 minutes or so. So
yeah,
let's keep the question.
Perfect. Yeah, it's a very short part. This is ongoing project, so just a very small taste of it. But hopefully I convince you there's a good reason to be able to program reference curvature and metric in structures. First, because we can control the geometrical frustration, such as I told you in the first project, and the Mainardi-Kodazzi frustration involves controlling both A bar and B bar. Other reasons we have is to build non-frustrated structures. In some cases, we would like the shape-morphing structures to adopt configurations which are unfrustrated. So for example, if they will break apart, they won't change the shape. Or there are other reasons. Another thing is that recently there are more and more works about programming mechanical properties, not only shapes, using the reference geometry. So we can program soft modes, we can tune nonlinearities and other properties in these materials. And in this, we try to utilize the fact that the fabrication techniques in the past 10 years got very, very advanced, and we try to use 3D printers here. And if we, again, try cartoonishly see what our ability to program materials is. So in the beginning, there was 1D control. So in these hydrogels I showed you before, the way people made them, just injecting solutions between two glass plates with a drill, hole in one of them. And then you can control just the radial profile of the gel composition. You get a disc that grows of the solution. And just by changing what you inject, you can change the radial composition of the gel. Then you can do a bit better and use lithography-like systems when you shine UV, for example, and then you can really have 2D control over your reference geometry. Then people started using these 3D printers when you really can now inject hydrogels with filaments, and these filaments are elongated or aligned along your printing directions, very similar to the thermoplastics I discussed before. But in here we ask ourselves, can we try to think of a way to really program fully the reference geometry of a material, even not necessarily a thin sheet, really programming the bulk properties of the material? And our idea was to use a 3D printer. So this is a DLP 3D printer. We use a projector that shines UV light in order to selectively crosslink and polymerize the hydrogel. So we just add layer by layer of this hydrogel. And we're working with two hydrogels, actually, polyacrylic acid of two different cross-linking densities. And the higher density, once we immerse the gel afterwards in water, will swell a bit, but the other one, the less cross-linked, will swell by a large factor. So by programming and playing with these two materials, we can make or program reference geometries. And the design idea is we have a desired structure and we have the desired geometry. We know by how much we want every pixel to swell. And instead of having a grayscale value, we just pixelize it. So we're just getting black and white pixels at high enough resolution that elasticity will smooth it out into a smooth gradients in composition, very similar to how our eyes smooth out black and white things we print in a printer. And so what we do, we just print one layer of the first material, we print the other material, and then the second layer and the third layer, and so on. In this case, now we'll focus on two layers because, as I told you, that to program A bar and B bar, you only need two layers of your material. But in practice, you can use this technique to program also bulk materials, the reference geometry of bulk materials. And here is just an electron micrograph that shows you that we really get this pixelated structure. And the first thing in all these systems is trying to calibrate them. So getting a uniform pixel density, varying density. If the density is 35%, we don't get swelling at all. And we can get up to 20-25% of linear swelling, so each direction. So the area swells cubically with respect to that. Then we can have bilayers, so one layer with no dark pixels, and we can vary the amount of dark pixel at the bottom or top layer, controlling the direction of curvature. And here our result, we have no fitting parameters once we know this calibration curve. So we can now program both A bar and B bar. We can move on to program gradients. So we can program gradients in A bar just by having different local concentration of these pixels. And we can also control B bar. So here we see an example. We have a uniform average concentration of pixels, but they just move from being at the top to the bottom in a sinusoidal way. We can control the number of nodes. And these days, we now try to print more impressive things such as cars and faces. So hopefully, again, in a few weeks, even, we'll have some exciting results to publish. So that's it. I think I'm anyway almost out of time. So just a quick summary. I started by telling you about a new pathway to induce shape morphing in sheets. It can help us to control the scaling laws of the shape transition. Even in some cases, there is no shape transition. So we are very excited about that. Then about the first completely autonomous shape morphing sheet, these busy gels. So we try to teach them how to swim, and we look at them as minimal models for active solids. So these very complex biological systems that are very hard to study, maybe you can get insights looking at these at least more relatively simple systems. And lastly, very briefly, I told you about our new approach that we called 3D printing of digital metamaterials, that hopefully you'll hear more about soon. And again, just to acknowledge my collaborators. So everything happened in Eran Shoron's lab at the Hebrew University. The first project with Emmanuel and C, the busy gels with Robert Digan from the University of Michigan and Omri. And the last project took place at the Shlomo Magdassi laboratory at the Kazali Institute, again with Ella and Rama. And you can read more about the two projects in our papers, hopefully soon on the third project as well, and to acknowledge my funding. And thank you for listening.
Thank you, Ido. That was fantastic. So yeah, now I guess we can stop recording and take further questions. Yeah, everyone feel free to unmute yourself and ask questions directly.